Okay, so this presentation is in response to exam 1D, question number 10, concerning toll-like receptors. So let's start off by pointing out that toll-like receptors are a type of pattern recognition receptors. Pattern recognition receptors are receptors expressed by cells of the innate immune system, and their job is to essentially identify two classes of molecules. They need to be able to recognize the highly conserved structural motifs known as PAMPs, or pathogen-associated microbial patterns, or molecular patterns. And these are molecules that are exclusively expressed by microbial pathogens. And then you also have the DAMPs, or danger or damage-associated molecular patterns. And these are endogenous molecules that are going to be released by dead and dying cells. PAMPs, the pathogen-associated microbial patterns, can really be any of a number of things. They can include various bacterial cell wall components. Lipopolysaccharide seems to be the prototypical PAMP. Other lipopeptides, molecules like flagellin or pieces of bacterial DNA, double-stranded viral RNA. All of these things can serve as PAMPs that will trigger the toll-like receptor if a pathogen is present. DAMPs can include intracellular proteins, heat shock proteins, also protein fragments from the extracellular matrix. Stimulation of the toll-like receptor via the corresponding PAMP or DAMP will initiate a signaling cascade that leads to the activation of transcription factors, as well as interferon regulatory factors. Signaling by the toll-like receptors can result in a variety of cellular responses, including the production of interferons, the production of pro-inflammatory cytokines, and effector cytokines that will direct the adaptive immune response. So TLRs actually occur in a family. They're a type 1 transmembrane protein, and they're characterized by this extracellular domain containing leucine-rich repeats. These leucine-rich repeats essentially form a horseshoe shape, and then there's also a cytoplasmic tail that contains a conserved region called the TIR domain, the toll IL-1 receptor domain. TLRs are predominantly expressed in tissues involved in immune functions, such as the spleen or peripheral blood leukocytes, as well as those exposed to external environments such as lungs, the gastrointestinal tract. These things are bringing in outside materials. You're going to want those types of cells to be there. Their expression profiles vary from tissue to tissue and cell type to cell type. TLRs are located on the plasma membrane, with some exceptions. Number 3, number 7, and number 9 are localized in the endosomal compartment. So a number of TLRs have been identified, 10 human and 12 in mice. So 1 through 10 are found in humans. 1 through 9 are found in mice. A homologue of 10, a homologue of TLR10 that is found in humans is found in mice. And 11, 12, and 13 seem to be exclusively in the mouse model. TLRs 1, 2, and 6 are essential for the recognition of a variety of PAMPs from gram-positive bacteria. TLR3 is implicated in recognizing viral double-stranded RNA. TLR4 is predominantly activated by lipopolysaccharide, LPS. 5 detects bacterial flagellin. 7 and 8 recognize synthetic antiviral molecules. 9 is required for a response to unmethylated CpG DNA. 10 is still a mystery. We're still working on understanding TLR10 and its ligand. So once a TLR recognizes a PAMP and signaling begins, signaling consists of at least 2 distinct pathways. A MyD88 dependent pathway that leads to the production of inflammatory cytokines, and a MyD88 independent pathway associated with the stimulation of IFN beta and the maturation of dendritic cells. The MyD88 dependent pathway is common to all the toll-like receptors except for number 3. Upon activation by their associated PAMP or DAMP, TLRs will either hetero or homodimerize, inducing the recruitment of adapter proteins via the TIR domain. Adapter proteins include the TIR domain containing proteins, MyD88, TIR associated protein, MyD88 adapter-like protein, TIR domain containing adapter protein inducing IFN beta, and TRIF related adapter molecule. Individual toll-like receptors will induce different signaling responses by usage of the different adapter molecules. For example, TLR4 and 2 signaling requires the adapter TRIP MAL. This is involved in the MyD88 dependent pathway. TLR3, 7, 8, and 9 recognize viral nucleic acids and induce type 1 IFNs. The signaling mechanism leading to the induction of type 1 IFNs will differ depending on which TLR is activated. They involve the interferon regulatory factors, which are a family of transcription factors that are known to play a role in antiviral defense, cell growth, immune regulation. 3 factors, 3, 5, and 7, function as direct transducers of virus-mediated TLR signaling. TLR3 and 4 activate IRF3 and 7, while TLR 7 and 8 activate IRF 5 and 7. Thankfully, a significant progress has been made over the past few years in understanding TLR function. They are essential in host defense against pathogens. So although signaling is paramount in eradicating microbial infections and promoting tissue repair, it must also be tightly regulated. TLRs are implicated in a number of inflammatory and immune disorders and even play a role in cancer. So many single nucleotide polymorphisms have been identified in various TLR genes and are associated with particular diseases. Several therapeutic agents targeting those receptors are currently under preclinical and clinical evaluation. However, the complexity gets troublesome here. TLRs act as double-edged swords, you could say. They will either promote or inhibit disease progress. Therapeutic agents targeting those TLRs must be able to antagonize the harmful effects without affecting host defense functions. So there's definitely some hurdles to overcome here. Nonetheless, the potential of directing the innate immune system with drugs that can stimulate and activate and control the toll-like receptors to prevent and treat inflammatory and autoimmune diseases, it's certainly promising. The research is out there and underway.